Okay, so today's video is going to kind of wrap up all the different things about the nervous system. So we're going to talk about how it can elicit a response, and so a specific example of how the motor output here from the nervous system can elicit a response from the effector, and we are going to focus on how it elicits a muscle contraction. We're going to look at how it processes the information in the central nervous system, as well as some specialized receptors that allow all of these things to happen. Okay, so let's start first with the muscle contraction. Okay, so let's take a quick look at the anatomy of a muscle. And so, as many of y'all know, I teach anatomy. I taught anatomy in college. So, there's some times where I will kind of give you a little bit above and beyond. I'll try to filter that down a little bit for you for this particular situation. Okay, so let's do a little biology application here. Okay, and let's apply this to our, do a little review for our hierarchy here. Okay, so the muscle is going to be equivalent to an organ, right? And if y'all remember, an organ is a group of tissues that work together. Okay, so that means these have to be tissues. Okay, these bundles of muscle fibers, which are also called fascicles. Okay, so the fascicle is the bundle of muscle fibers. Those are the tissues. And the muscle itself is going to be made up of multiple fascicles. Okay, so going even smaller then, down from a tissue, okay, you're going to get down to the actual muscle cell. Okay, so now I'm at the cellular level. Okay, and the cell is going to be made up of these little guys here, which are called myofibrils. And we'll focus a lot on those, because those are going to be our proteins that are going to actually contract. Our actin, and our myosin that are going to actually contract and shorten and allow for that muscle to actually have some movement. So zooming in even further then into, you can see it's pointing out these myofibrils right here. Zooming in even further into what's called the sarcomere. Okay, and so the sarcomere is what we would call the functional unit of the muscle. This is where the actual contraction is taking place. Okay, so we've got tons and tons of these sarcomeres. You can see a sarcomere is going to run from what's called a Z line or a Z disc. Okay, you'll hear me call it a Z disc pretty often. It's the same thing. So sarcomeres run from Z line to Z line. So you'll notice right here along this muscle cell, I've got multiple sarcomeres. And so again, these are the functional unit of the muscle cell. This is what's going to actually contract. And so this sarcomere is going to consist of a variety of areas. So I've got what's called the I band here. And so that spans two sarcomeres. Because remember, a sarcomere runs from Z disc, okay, to Z disc. So the I band spans two sarcomeres. Okay, it runs, the Z disc runs right in between it. The A band here is right in the middle. And so the A band contains both actin and myosin. Okay, you can see here I've got my thick filaments versus my thin filaments. My thick filaments are these purpley looking ones here. That's the myosin. The thin filaments are the actin here. So in this I band, okay, I'm actin only. It's also sometimes called the light band. Okay, and so in the A band, then I'm actin and myosin, and so it's sometimes called the dark band. Okay, when you look at skeletal muscle cells under the microscope, the black and white picture right here shows us that. Okay, you see I've got these dark bands, the A band, versus the light band, which is the I band. Okay, so in addition to the bands and the actin and myosin, we have this H zone right here in the middle, which is also sometimes called, let's come way over here, okay, it's also sometimes called the Baer zone because it has no actin. This is what actually allows for muscle contraction. Okay, when the muscle contracts, the actin is going to move in towards the middle like this. And so we need this space in the middle that doesn't have any actin, so it has somewhere to go. You can see this one, the black and white one here that's from the microscope, okay, here is contracted. That H zone or Baer zone doesn't really exist because the actin has been pulled in towards the middle. Okay. So this is our sarcomere. Like I said, this is our functional unit. This is what's going to actually contract. So let's look at what happens when it contracts. Okay, so when the muscle actually contracts, okay, the sarcomere is going to shorten. So over here on the left, I've got a muscle that is relaxed. So I've got that empty H zone or that bare zone. And over here on the right, I've got a muscle that's contracted. And so that actin has moved in there towards the middle. Okay, that line running right down through the middle is called the M line. And so it has moved in towards the middle, towards that M line. But you'll notice they haven't changed shape. The actin and the myosin here, so here's my myosin, the purple, the actin in the gold, okay? They haven't changed shape. They have not actually gotten any shorter. They've just been pulled closer together. Okay, so it's just a position change, which is called our sliding filament theory. That these filaments, these myofibrils, okay, these protein filaments, slide over one another as the muscle contracts. Okay, so let's take a look at what's required to do this. You're going to need some of your ions. You're going to need a signal from the nervous system to actually make this contract. Okay, so we're going to look at this diagram probably a couple of times, but I want you to get an overview before we start to get into our get into a little bit more detail. So if you'll notice, what I have here is that my myosin, okay, is going to eventually bond to my actin, and when my myosin bonds to the actin, okay, you'll notice the head of the myosin here changes position, okay, and so it essentially grabs a hold of the actin and pulls it in towards the middle. And then it will release, okay, and then it will grab it again, and it will pull it towards the middle again. Okay. The only problem we have is that the myosin actually loves the actin, would love to be attached to it all the time, but the actin, the binding site on the actin, is covered up. And it's covered up by something called the troponin-tropomyosin complex. So we need to get that out of the way before this actual contraction can take place. Okay, so at rest, the muscle cell, the binding sites on the actin, you can see them here, these kind of dark-looking circles here. Okay, those binding sites on the actin are covered up. And they're covered up by this filament here that's called tropomyosin. Okay, and on this tropomyosin complex, you'll have the troponin protein. And the troponin protein will pull that tropomyosin out of the way so that the myosin can actually bind to the actin. Okay? And it will do that in the presence of calcium. And if y'all remember from when we talked about cell communication and stuff, calcium is not usually present in the cytoplasm. It's usually stored either outside of the cell or it's stored in the endoplasmic reticulum. And muscle cells have a special endoplasmic reticulum called the sarcoplasmic reticulum. And like you said, it's just a specialized endoplasmic reticulum where the calcium will be stored. So when the sarcoplasmic reticulum releases the calcium out into the muscle cell, that calcium will bind to the troponin complex. And when it binds to the troponin complex, it pulls that tropomyosin out of the way, and now these myosin binding sites are free and open. And so the myosin head can now actually bind to the actin so that the contraction can happen. So coming back to our overview here, so in this picture, the calcium has to have bound already to the troponin complex to move the tropomyosin out of the way. And so what we have here is these free myosin binding sites on the actin, and so the myosin is going to bind to it. And so when the myosin head is in what's called a high energy configuration, so that requires the breakdown of ATP. So ATP has to be present for muscle contraction to happen. Okay, remember mitochondria make ATP. Muscle cells have a very high concentration of mitochondria because they require a significant amount of ATP to make muscles contract. So you're going to see an increase in a spike in the concentration of mitochondria to help keep up with this demand. Okay, so when the phosphate is cleaved off of the ATP, and now it's an ADP, the myosin head reaches what's called a high energy configuration. And in that high energy configuration, it can form the cross bridge. And so the cross bridge is when the actin and the myosin are actually bound together. And that triggers, when that cross bridge is formed, that triggers the release of the ADP molecule. And when the ADP molecule is released, the myosin head goes back to its low energy configuration. And if you notice the angle of the head, okay, in the low energy configuration, it's angled more towards the center. Where in the in the low angled configuration, it's like this in the low energy configuration. In the high energy configuration, it's more straight up and down. So when it reverts from this low energy configuration, it moves this direction, which causes the actin to be pulled towards the middle. So when this ADP is released from the myosin head and it returns back to the low energy configuration, it's going to pull that actin towards the middle. And when another ATP comes back, okay, and that ATP binds to the myosin head, it will release and go back. Okay? It will release so that it can form its high energy configuration again. And so the myosin heads will continuously bind, form the cross bridge, pull, bind, pull, bind, pull, okay, and move that actin more towards the middle. Slide that actin, doing that sliding filament theory, more towards the center of the sarcomere, which causes the whole sarcomere to shorten up. So it requires ATP actually to relax and release the myosin head from the actin. Okay, I want you to kind of think about for a little bit, see if you can figure out the answer as to why rigor mortis would occur then. Okay, based on what you now know about what's required of it for a muscle cell to contract, okay, why would rigor mortis actually happen? And for those of you that don't know what that is when the dead body stays stiff. Okay, its muscles are almost all contracted and it's stiff. So just think about that. Okay.
Skeletal muscle is composed of bundles of elongated muscle cells called fibers. Each muscle fiber or cell contains thread-like structures called myofibrils. A myofibril is made of units called sarcomeres, joined end-to-end by Z-lines. Each sarcomere contains both thin and thick strands of proteins known as myofilaments. The thin myofilaments are attached to the Z-lines at both ends. Thick filaments are grouped in the sarcomere's center. When thin and thick filaments interact, they slide past one another. This sliding action pulls the Z-lines toward the center, and the sarcomere shortens. As the sarcomere shortens, the entire muscle shortens or contracts. Thick myofilaments are made of the protein myosin. Individual myosin molecules have